CRISPR-Cas9 system. What is CRISPR-Cas9? In 1987, scientists identified the first signs that prokaryotes had an undiscovered immune system that could remember previous infections. The first clue was an unusual region in the prokaryotic genome that was later called clustered regularly interspaced short palindromic repeats, CRISPR, pronounced CRISPR, that had a strange DNA sequence. By 2007, it was realized that this sequence was the basis of a prokaryotic immune system. It not only enabled microbes to remember infections so they could react more quickly, but they could also pass this knowledge on to their offspring. It eventually became apparent that CRISPR-Cas9 was functionally equivalent to our adaptive immune system. CRISPR-Cas9 helps to protect prokaryotes from viral and plasmid infection. When a virus infects a cell, a viral DNA genome is chopped into short fragments by CRISPR-associated enzymes, and these viral DNA fragments are integrated into the prokaryotic genome at the CRISPR locus. This whole locus is transcribed into a long non-coding RNA that is in turn cut into smaller guide RNAs, each with a small section of viral DNA. The guide RNAs are taken up by enzymes such as Cas9, so they can hunt for additional copies of the viral DNA. When found, the viral DNA copies are chopped up, stopping the viral infection. CRISPR-Cas9 is a scientific tool. This knowledge of CRISPR-Cas9 has enabled the development of a genome editing system that has become a very powerful tool for research scientists and may lead to new ways to treat many illnesses. Scientists have adapted CRISPR-Cas9 into a precise targeting system to edit genomes or make other changes to DNA that were previously time-consuming and expensive. As it is easy to create artificial guide RNAs to target specific places in a genome, a scientist can use CRISPR-Cas9 to permanently disable a gene, temporarily turn it on or off, or permanently edit a region in a genome with precision. Before looking in more detail at how CRISPR-Cas9 is used, we will just consider how it works. So how does it work? Cas9 is the DNA cutting enzyme. It's a nuclease that is used in CRISPR-Cas9 genome editing. Cas9 will find and cut the target DNA. X-ray crystallography has shown that Cas9 has two main lobes with different functions, a recognition lobe and a nuclease lobe. The recognition lobe finds the genome target by using a guide RNA that is complementary to the genome target. The guide RNA has a short sequence that allows it to interact with Cas9, and then a 20 base target sequence that will vary depending on the target. Thus, different targets will need different guide RNAs, but the same Cas9 enzyme can be used. When Cas9 binds the guide RNA, it changes shape, forming a central channel, where it stretches out the guide RNA so it can interact with complementary DNA bases when genomic DNA is also in the channel. Cas9 can only bind DNA when it has a guide RNA associated with it. The Cas9 nuclease lobe has two nuclease domains that can each cut one strand of the genomic DNA. When Cas9 in a complex with guide RNA binds the matching DNA target, it undergoes a conformational change, so the nuclease domains cut both strands of DNA. The nuclease lobe is also interacting with the PAM sequence that is important for CRISPR-Cas9. PAM sequence. PAM is a PAM stands for protospacer adjacent motif sequence. Is how prokaryotes avoid cutting their own genomes up with their CRISPR-Cas9 system. The PAM sequence is a short sequence of bases that doesn't occur in the prokaryote's own genome. For example, 5' NGG3 is used by the most commonly used Cas9 for genome editing. It occurs by chance about every 10 base pairs in human genome. PAM is essential for Cas9 binding to the DNA genome. Cas9 cuts 3 or 4 bases away from the PAM sequence. Two different systems in cells will repair the DNA breaks. One is more efficient but error-prone, NHEJ, and the other very accurate but less active, HDR, so only used for 10% of repairs. The CRISPR-Cas9 system exploits the cell's own DNA repair systems to fix the double-stranded break to give the outcome the researcher wants. So gene knockout with CRISPR-Cas9. One way to understand what a gene does is to see what happens when the gene is removed. Finding a mutant or deactivating a gene knockout, is a time-consuming and expensive process that CRISPR-Cas9 has made much easier. To knock out a gene with CRISPR-Cas9, you need to design a guide RNA that has a unique target and is just upstream of a PAM sequence. Cas9 with a guide RNA will bind to the PAM sequence anywhere in the genome, but only cuts the DNA if the guide RNA target sequence matches the DNA genome target. When it finds such a match, Cas9 makes a double-stranded DNA break. In a gene knockout, the NHEJ DNA repair system will fixes the break by adding or removing a few nucleotide bases at that break. These bases often cause a mutation, such as a frame shift, that disables the gene. Obviously, the knockout effects will vary depending on how the repair was made. Genome editing with CRISPR-Cas9. One of the most powerful and to some most frightening uses of CRISPR-Cas9 is to edit a genome. Scientists can change one base or add a whole gene, so it really is a very flexible system. Crucially, such changes can be passed on to future generations. To do this, scientists must provide a DNA repair template with the new DNA sequence that has a matching or homologous sequence upstream and downstream of the genome target. Furthermore, the repair template must not have a PAM sequence or it will also be cut by Cas9. Genome editing uses the inefficient HTR DNA repair system, so less than 10% of Cas9 DNA cuts are repaired as desired. Thus, it is necessary to be able to identify cells with correctly altered DNA from the incorrect ones. Regardless of this necessity, CRISPR-Cas9 still offers a very simple way to edit genomes. Controlling gene transcription with CRISPR-Cas9, something else it can do. As Cas9 binds to a genome target independently of cutting it, it can be used to accurately find specific sites in the genome. By deactivating both Cas9 nuclease domains, the protein will find a target and bind, but not cut it. So scientists have found that by adding a transcriptional regulating protein subunit to a Cas9 with active nucleases, you can use it to switch specific genes on or off when the genome target is a promoter. Some applications of CRISPR being used now. Therapeutic applications. Gene therapy. CRISPR-based gene therapies are being developed for a range of genetic disorders: sickle cell disease, beta thalassemia, and Duchenne muscular dystrophy. Cancer immunotherapy. CRISPR is being used to engineer T cells and NK cells to target and destroy cancer cells, with trials underway for various cancers including leukemia, lymphoma, and non-small cell lung cancer. Viral infection treatment. CRISPR is being explored as a potential treatment for viral infections like HIV by targeting and eliminating the virus from infected cells. Hematologic diseases. CRISPR-Cas9 is being used to correct genetic errors in hematopoietic stem cells that cause hematologic diseases, with promising results in treating sickle cell disease. Clinical trials. There's ongoing trials. Numerous clinical trials are underway, focusing on various applications of CRISPR technology, including cancer immunotherapy, gene therapy for genetic disorders, and treatment for viral infections. There's FDA approvals. The FDA has approved the first CRISPR treatment for sickle cell disease, Cas-GEVY, and Excision Bio's EBT-101 therapy for HIV has been granted fast track status by the FDA in its phase 1-2 clinical trials, and is in phase 1-2 clinical trials. Agriculture applications. Climate change resilience. CRISPR is used to engineer crops that are more resistant to drought, heat, and other climate-related stressors. Pest and disease resistance. CRISPR can create crops that are naturally resistant to pests and diseases, reducing the need for pesticides. And then improved crop yields. CRISPR can be used to enhance crop yields and improve the nutritional value of food.